I'm going to try and explain the protein folding process. Most of my roommates are either away or in bed, so I guess I'm just going to have to talk to the camera by myself. Before folding can start, you need something to fold, which in case of the protein folding process, you need a linear sequence of amino acids, which we call the primary structure. And the folding process begins when local or amino acids within close proximity of each other start to interact. As the folding process continues, more complex structures are created, the next being secondary structures, which contain hydrogen bonds, alpha helicases, and beta sheets. And assuming the folding process continues, the hydrophobic effect will occur, which makes hydrophobic amino acids cluster together to avoid water from the hydrogen bonds, which will result in a hydrophobic collapse. The next structure we have is the tertiary structure, which is a three-dimensional structure formed by long-range interactions. So farther apart amino acids in the chains are now interacting with each other to create all sorts of weak bonds, so ionic bonds, dispersion forces, and disulfide bonds. So there's a lot of versatility in the tertiary structure. Next structure we have is the quaternary structure, which is not as common as the other three structures, but basically what it is, it's multiple polypeptide chains formed together, and assuming everything is all right, the protein will be now in its native state, which is the lowest energy state that is required to hold that position, and that will be at the bottom of the energy landscape. And the process to getting to that native state is the protein going through intermediates. So instead of the protein going from like primary straight to secondary, there will be intermediates between those structures to help with the folding process, along with chaperones, which help stabilize the proteins so that they don't misfold and aggregate, which will, if that happens, it will cause diseases such as Alzheimer's and Parkinson's.